The overall goal of implementing online size exclusion and ion exchange chromatography on small-angle X-ray scattering beamlines is to obtain better quality low-resolution model data by ensuring minimal delays between sample preparation and data acquisition.
These methods can help to answer key questions in the field of structural biology, like the radius of gyration, the largest intraparticular distance, the particle shape, the degree of folding, denaturation or disorder.
SAXS requires monodisperse samples. The main advantage of these techniques is that the time between the purification and the measurement is short, helping to avoid sample denaturation or aggregation.
Before going to the Beamline facility, prepare a concentrated sample using previously published methods and perform SEC test runs. At the Beamline facility, place 1 ml of gel filtration buffer in a 1.5 ml reaction tube. Then, connect a bottle of gel filtration buffer to an HPLC system in line with the flow-through capillary of the SAXS setup. Set the flow rate, maximum column pressure, and acquisition time. Note the back pressure without the column. Purge the lines and then connect and equilibrate the size-exclusion chromatography column. Load the 1 ml buffer sample into the sample changer. Exit and secure or interlock the experimental hutch. Then, log into the Beamline control software in sample changer mode. Send the buffer sample in the sample changer directly through the SAXS flow-through capillary. Perform a short SAXS test run and